Good morning, good afternoon, good evening to all of you joining us from around the world today. This webinar is entitled Biodegradable Long-Acting Injectable Implants Prepared by Hot Melt Extrusion. This webinar has been made possible by Thermo Fisher Scientific, so a big thanks to them for enabling us to gather today and share science together. Our speaker today is Dr. Tan Nguyen, Product Manager at Pharmaceuticals. His presentation today will focus on injectable implants using twin screw extrusion, an exciting new technology in pharmaceutical manufacturing. My name is Darius Sulam from the events team at Inside Scientific, and I'm pleased to be your host today. Now, just a few housekeeping notes before getting started. This webinar is being recorded and resources will be made available following the event. If the webinar panels look too big or too small, you can zoom in or out in your internet browser to adjust the viewing area. You can also resize some of these panels or make the media panel full screen. Just before getting started, I would like to take a moment to run a few audience polls. The first question, are you familiar with hot melt extrusion? Yes or no? While you're answering this question, I'd like to draw your attention to the resources panel on the top right of your screen, where you'll find a few links and suggested readings associated with today's event. Thanks very much for participating in this question. I'm going to move now to the next polling question. Do you work with biodegradable polymers? Yes or no? Now, while you're answering this question, I'll also draw your attention to the panel on the bottom right, which is the Ask a Question box. At any point, if you have questions, thoughts, and comments, use this box next to the media window at any time throughout the presentation, and your questions will be addressed during the Q&A session after the presentation. Thanks so much for answering this question. We're going to move to the final polling question. Do you have experience with injectable implants already? Yes or no? If you happen to experience any technical issues during the event, the easy fix tends to be a simple refresh of the webinar auditorium page by refreshing your browser. This should successfully reestablish your streaming connection so you can hear and see us clearly. However, if this doesn't work and you continue to have issues, just use the Ask a Question box to communicate your issue with our team, and we will help you to get back up and running. Thanks so much for participating in those polls. At this time, I'm going to bring Tan Nguyen onto the scene. Tan, thank you for being here.
Thank you, Darius, for the introduction. So hello, everyone. Today, I would like to share with you our experiences in the development of biodegradable, long-acting injectable implants prepared by the hot melt extrusion. But first of all, I would like to introduce myself. So my name is Tan Nguyen, and I'm a program manager at Pharmaceutical, and I am responsible for all the projects related to the development of long-acting injectable that we can prepare by hot melt extrusion or by in situ forming gel. And the presentation today will focus on hot melt extrusion as a platform technology. So Innocore Pharmaceutical, in a few words, we are drug delivery company. We are located in the north of the Netherlands. It's about 2 hours from Amsterdam, and we are a team of about 40 employees, 40 colleagues, with more than 75% having a Master of Science or PhD degree. So we really focus on development of injectable sustainreleased drug delivery, and in our portfolio we have our patented polymer base for the drug delivery, and we developed the project until the GMP manufacturing and supply for the phase 1, phase 2 clinical study. We work in a partnership with different big pharma and small biotech, and we work in a license and development business model. So why are we working on the development of sustained-release drug delivery system? Clearly, there's some significant benefit for the patient and also for the healthcare system as well. If you look at the graph on the in this slide, you can see that it's there's drug plasma concentration curve in purple that show you the variation of the drug concentration in the blood, and it may go over the limit to reach a toxic level, or it may also go under the level that you can have the efficacy, the therapeutic efficacy of the treatment. That is a drawback of immediate release, and advantage of the sustained release or long-acting injectable is that we can deliver a stable drug concentration in plasma at within the therapeutic range, meaning lower than the toxic level, but also higher than the minimum effective level. And to illustrate the benefit of long-acting injectable, we have a few examples, and the first one is if we look at some application where you need to inject the drug in the eye. So clearly you can see the big difference between a daily injection to the eye, or if you have a long-acting injectable, you only need to inject once a month, or even once every 6 months, as one of the products we are developing at this moment in partnership with AbbVie and Allergan. That will bring a significant convenient benefit to the patient, and the reduce in the injection number will in the number of injection will also decrease the risk of inflammation and the risk of infection to the patient associated with each injection. So in the second example is about the side effect of the drug. One very clear example here is exenatide as an API that was hardly known as an API for the treatment of diabetes before the appearance of Bydureon and a long-acting injectable formulation that would help to reduce the side effect of the patient. A third example is the compliance in psychiatry. You can imagine what would happen if a patient just forgot to take their medication and became violent. And the last one, in the area of family planning, what would happen if we have incompliance? So at Innoco, we developed the long-acting injectable with using different technology platform. We produce a formulation with microparticles or solid implants or drug-eluting coating or injectable gels. We have our Symbiosis multi-block copolymer platform and also in-gel tri-block copolymers to develop the long-acting injectable. And for today, I will focus on the solid implant using Symbiosis polymer and hot melt extrusion. Just a little bit over the business model. We normally would start a project with a client or a partner with the API and application provided by the partner, and we provide we contribute with our drug delivery system, and we go all the way from R&D feasibility study to GLP tox study, GMP manufacturing and supply for the phase 1-2 clinical study. And when it comes to the late stage manufacturing for clinical trial and commercial manufacturing, we will just hand it back to either partner or our selected CMO for the manufacturing. So we keep our core expertise in the R&D and early development to the phase 1-2 clinical study. Now, let's have a look at the long-acting injectable solid implants. If you look at the long-acting injectable implants available in the market, we saw a growth of non-biodegradable implants such as Norplan, Levoplan, Implanon, Jadel. These are the implants that are used for long-term efficacy, say 3 to 5 years, and these are pretty big implant that contains also high amount of active. These are non-biodegradable. There are also other non-biodegradable implants that were used for the treatment of prostate cancer, and these implants also contain a high amount of API. In the same therapeutic area for prostate cancer treatment, there were quite famous product marketed by AstraZeneca. These are Zoladex that contain goserelin that can be used for the treatment of late-stage prostate cancer. These are biodegradable implants, and there are in the category of biodegradable, we can also see Oxudex, which is developed and marketed by Allergan. These are the tiny. This is a tiny implant that will be delivered to the eye for treatment of inflammation. And recently, we saw a new marketing approval of another product with biodegradable implant that is used for the treatment of phototoxic. So it is toxicity due to different sources of light. Here in this table, we provide a summaries of different long-acting injectable implant available in the market, and you can see that the latest one was used for the treatment of erythropoietic protoporphyria, which was approved in 2019, and this is also biodegradable implant that will provide 2 months of treatment duration. Now, at Innocore, we developed the long-acting injectable solid implant using hot melt extrusion as both the monolithic matrix and in the form of the implant with the release controlling layer. But for monolithic matrix, we use different extruder, the small Hacker Mini Lab extruder for very small scale early development at about 5 to 7 grams of material. And once we have a lead formulation, we can scale up to clinical manufacturing, to manufacturing at a clinical supply scale or even a commercial scale with an 11 millimeter extruder. So in the top left picture, you can see a simplified manufacturing process. In principle, we just need to compound the polymer or blend the polymer and the API and introduce the blend in a hot melt extruder to melt and mix the components, and then create a shape of the implant via a die, a rod die, and then we cool down the material and cut into implant. So yeah, when we talk about implant, we normally think about a cylindrical rod, but we also develop the implant in the form of a thin sheet as well. So yeah, I would like to introduce or share with you our experiences in the development of a generic formulation or generic product of Zoladex. But Zoladex is goserelin implant product which are available in two strengths. The first one contains 3.6 milligram of goserelin, and which is indicated for 1 month treatment of prostate cancer, while the second strand contains 3 times higher than the amount of the drug, the API, 10.8 milligram of goserelin, and which is used for 3 months treatment of prostate cancer. And the two strand has different dimension. The implant of the two strands has different dimension and will be delivered by two different injector with different needle size. The 16G needle for the 1 month and 14G needle for the 3 months. So at Innocore, we developed a generic product of goserelin implant, which we call implant. And in the lab, we develop the formulation, the implant formulation, and compare the drug release characteristic by in vitro release experiment. And we can see that you can see that here in the solid green line is the cumulative release in vitro of the generic formulation, the implant, and the dotted green line is a release profile of the originator, Zoladex 3.6 milligram for 1 month release. So and the two dotted black curve are the specification indicated in the British Pharmacopoeia. So if you are within these two black curve, and you are within specification of the Pharmacopoeia. And you can see here that the release profile of the 1 month sustained release formulation is really typical of a PLGA polymer matrix, which is non-linear and biphasic. In the first week, so from day 0 to 7, we have very slow release, and once the polymer started to degrade, then we will have an increase in the drug release rate from day 7 to day 21. You can see, and when most of the drug content from the implant have been released, we'll have another decrease in the release rate when the sources of API inside the implant are are running low. So next to the in vitro characterization, we also characterize or compare the originator with our generic implant in vivo in rat model, and we characterize in both pharmacokinetic and pharmacodynamic, which is the efficacy of the API. So in the left y-axis, you can see the scale of the serum goserelin concentration in nanogram per milliliter. So it is expressed in the red curve. So in vivo, the release profile of the generic implant and the originator implant are almost the same. So they are very similar, and that's so that the generic implant has met the requirement for the in vitro character for the in vivo pharmacokinetic. But on top of that, if we look at the pharmacodynamic, which is the efficacy of the API, we see the same behavior of the generic implant and the originator implant. We saw normally an increase in the serum testosterone at the beginning after administration of the implant, but right after that, when we have a feedback loop, testosterone level will decrease to normal range of concentration, as you can see in the y-axis on the right side, serum testosterone in nanogram per milliliter. So the in vivo response to both generic implant and originator implant are quite similar. But what we can also notice from the pharmacokinetic in vivo is that the plasma concentration of goserelin from day 7 to day 21, we saw a significant increase in the concentration, but the response remained quite similar as compared to the area when we have a lower concentration of goserelin in the serum. So we could also conclude that here what we see is that between the day 7 and day 21, the goserelin was released more than needed to have the therapeutic effect. This is typical characteristic of PLCA polymer matrix. So again, when we put together the drug release from the in vitro characterization, which is expressed in the as a green curve, and with using the graph on the right side, the y-axis on the right side, and the in vivo is as a red curve, we can see that the increase in the serum goserelin is associated with the degradation phase of the polymer, and which is the real reason behind that increase in the drug concentration in plasma. Now, next to the development of 1-month formulation, we also developed a generic version of Zoladex implant. So we call it Implacres 3 months. So in vitro characterization also show similarity in the release profile of the generic formulation as compared to the originator, and both release profile were within the specification as defined in the British Pharmacopoeia. And again, due to the use of PLGA in the polymer matrix, we can see a typical degradation control release profile. As we can see from day 0 to about day 35, 38, we have a very slow release, and once the polymer started to degrade, so we have degradation control release characteristic with an increase in the release rate from day 38 to about day 30, and that would typically result in a spike in the plasma concentration of the drug, which is probably not necessary. And when we look at all the long-acting LSRS agonists that are available in the market, we saw a different version of the long-acting injectable, which has a different duration from 1 month, 3 months to 4, 6, and 12 months. But only in the case of Zoladex implant, we can only see 1 month and 3 months implant. We just cannot see. We do not cannot find 4, 6, or 12-month formulation. And we were thinking a lot about the reason behind that. Why can't we just have a longer acting implant to even reduce the administration frequency for the patient? The main reason probably was that if we need to increase the release duration, then we will need to increase the amount of API, so the amount of drug. So here, with a linear calculation, if we want to go to 4 months or 6 months, we will need 14 milligram. We need to deliver 14.6 milligram or 21.6 milligram of goserelin respectively. And with that amount of active, we need to have a bigger implant. So if we look at the 1 month implant, it's smaller implant with 1.2 millimeter in diameter and 10 millimeter in length. And when we increase the duration to 3 months, we need to use a bigger implant with 1.5 millimeter in diameter and 15 millimeter in length. Now, for the 4 months and 6 months, we just calculated the dimension, and we will see that it will be much. We will need a much bigger diameter than 1.5 millimeter. So imagine that an 1.5 millimeter diameter implant to be injected in the body. You will need to deliver the implant under anesthesia, and that is the downside of a larger implant administration. However, we do see that there are opportunity to deliver a longer sustained release of goserelin without having to increase the dimension of the implant, and which is still affordable for the patient. So here we just identify some disadvantages of the PLGA copolymer for long-acting injectable formulation development. Is that it's the irregular biphasic release kinetic of the polymer, which is the main drawback of the that platform. And we have already seen earlier that there was a spike in the plasma drug concentration, which is not really necessary to maintain the pharmacodynamics, so the efficacy of the drug. The second disadvantage of PLGA copolymer is that it will normally lead to a decrease in the EC2 PS, which could be destructive for some certain API, especially when we talk about the peptide or protein or antibody. And there are also other disadvantages related to adsorption of the peptide or protein on the hydrophobic surface of PLGA, or acylation of the API with the carboxylic acid end group of the PLGA. So all this would indicate that there is a need for alternative biodegradable polymer to develop the long-acting injectable implant with more constant drug release, and that can also address the drawback of the current PLGA in the formulation of in long-acting injectable implant development. So at Innocore, we develop and we have our intellectual property on another type of biodegradable polymer, which we call it the Symbiosis polymer. It is a hydrophilic biodegradable polyether ester urethane polymers, which composed of the well-known monomers like epsilon caprolactone or dialsanone or lactide or glycolide. These are all well-known monomer, and we synthesize the multi-block copolymer by first synthesizing a short block that we call pre-polymer, and then the different pre-polymer would be chain extended to combine together to form a multi-block copolymer with the unique molecular architecture that we can fine-tune to address the need of drug delivery. So either it is a small molecule or it is a peptide or protein. We can customize the polymer structure and characteristic to meet with the requirement to deliver the API. As I mentioned earlier, the monomer are well known. That could be either glycolide, lactide, caprolactone, dialsanone, and we also use known compound for as initiator like polyethylene glycol, butanediol, and tin octanoate as catalyst, and butan diisocyanate as a chain extender. All these components of the the Symbiosis block copolymer are well known, and these components are clinically proven as a safe polymer and chemicals with an excellent track record in the regulatory approval and in the marketed biomedical devices. Now, when we talk about the disadvantages of the PLA, PLGA polymer platform, and here we are talking about Symbiosis polymer and the advantages that it can address the current challenges in the drug delivery, long-acting injectable development. So the Symbiosis polymer is a multi-block copolymer with a phase-separated morphology. So there are two different phases: a hydrophilic amorphous domain, so phase of the polymer, that are alternating the hydrophobic crystalline domain or phases of the polymer, and this combination would make an unique structure of the Symbiosis polymer that can be used for the development of long-acting injectable. So the hydrophilic amorphous domain would actually absorb the water and swell to form hydrogel-like structure that allow the drug or the API to diffuse through the matrix. So that will be a diffusion control mechanism. And we can also control for the amount of water absorption or the swellability of the polymer based on the specific design of the polymer structure. And the other phase of the polymer, which is crystalline domain, that would maintain it's a cross-link domain that provides the structure integrity for the polymer or for the dosage form. The polymer will ultimately degrade through hydrolysis, and one unique characteristic of the polymer is that there's no accumulation of the acidic degradation product in situ, because the polymer can swell and the degradation product can diffuse through the hydrophilic amorphous domain. and that is really an unique distinction between Symbiosis polymer as compared to the conventional PLA, PLGA polymer. Here in this slide, we'd like to show that the Symbiosis polymer are well biocompatible, and that has all the safe degradation products that have been extensively tested and evaluated in vitro and in vivo in different animal model. And we also have an extensive ISO 10993 biocompatibility and toxicity data package that we can share with our partner during the development of a long-acting injectable. Again, on the possibility to customize the structure of Symbiosis polymer to address the need for long-acting for sustained release delivery, we can control the polymer in terms of swelling degree or degradation rate, and that would deliver different release rate and release profile. As you can see on the graph, there are 4 different polymer with different structure here, and we use the ratio between the hydrophilic block and hydrophobic crystalline block as 10-90, 20-80, up 30-70, and 50-50. And you can see in the release graph of a model API, which is a protein, the bovine serum albumin. We can see that the release profile can be fine-tuned or can be tailored by using different block ratio, leading to different release rate and release duration. So in a bit more detail, how to fine-tune the polymer, we can play with the molecular weight of the polyethylene glycol, or the content in percentage of polyethylene glycol, and the type of the monomer, weight fraction of monomer, and the block ratio, and the molecular weight of the multi-block copolymer as well. So there are different knobs that we can turn to fine-tune the release characteristic. Coming back to the example about gossamerine implant that I mentioned earlier, when we put together the pharmacokinetic in vivo as a serum gossamerine and the in vitro release characteristic in the green curve, so these are the in vitro data. So we can see that the increase in serum gossamerine concentration really correspond to the degradation of the PLGA polymer matrix between the day 7 and day 21, and that degradation would cause an unnecessary increase in the gossamerine concentration in plasma, which still can keep the efficacy of the drug, but this is probably not necessary. So there are possibility to reduce the dose by using different release mechanism. And based on that observation, we started with the development of a 4-month sustained release gossamerine implant, and we use our Symbiosis polymer based formulation. And you can see here on in the graph on the left side the in vitro release characterization of the 1 month in the green curve, the 3 month formulation in the blue curve, and the 4 months our inocore formulation as a red curve. You can see that the release of the API is more regular as compared to the PLGA matrix. And using the same dose, if we calculate the daily drug release from the implant using the same dose of 10.8 milligram of API, we can see that for the 1 month and the 3 month formulation, we will always have a spike in the daily release of goserelin. But for the Symbiosis based formulation, we see regular, quite constant daily release of goserelin for 4 months, which means that we could still develop a longer release product using the same amount of API, so without the need to increase the size of the implant, which would keep the same convenience level for the patient. In the previous example I showed you was the development of goserelin implant using a monolithic structure. But in some cases, we also need to develop dual layer or a two-layer product for providing an addition release controlling mechanism to the long-acting injectable implant. And at Innocore, we developed the dual layer implant either by using co-extrusion process, which is also well known for the manufacturing of Nuvaring or the Implano. But for some application, when we have the incompatibility between the polymer and in the core and the polymer in the membranes, either because of the degradation temperature of the API or the polymer, then we can use another process to produce a dual layer structure in a process what we call a wire coating process. And here in the video, you can see an example where we develop the outer layer that contain an API and that release the drug within 1 to 3 weeks, up to a month. So you can see here is an 11 millimeter hot melt extruder that we couple with a wire coating die, and on the one end of the die we have the core material coming in. So here is the material of the coating layer going into the extruder, and that will go to hot melt extrusion process. This is a conveyor belt. Also, we have inline laser control as a PAT control for the diameter of the implant and also the coating layer. Here you can see the wire coming in one side, and we'll meet the extruder with the coating material in the middle, and on the other side we have a coated tubing that we can monitor for the diameter using a laser, and then that will go out as a cylindrical tubing with a coated layer, and we can cut in the dimension that we want. So about the polymer, here we also demonstrate that we can use 3 different polymer grade with different amount content of the polyethylene glycol, which will provide different swelling degree. And there in the release characteristic, you can see the in vitro release profile of the API. We can have an almost immediate release, complete release within a day, or we can go to about 5 days to up to 2 weeks to 3 weeks sustained release of the API based on different structure of the polymer. Now, another area where we can use Symbiosis polymer, and we are actively working on it, is the development of long-acting reversible contraceptive. And there are different methods for contraceptive using around the world, but long-acting reversible contraceptive is up to now by far the most effective birth control method. Um, there are products available in the market, which was made of biodegradable, which means non-biodegradable polymer. But these are highly effective, but this implant always will require surgical removal of the implant after the treatment period. So we look again here at some product available in the market as a Norplan or Implanon or Jadiel. These are all very big implant, non-biodegradable. We would need to insert the implant in the patient under anesthesia, and also would require the surgical removal of the implant after the treatment duration as well. And at Innocore, we are developing the contraceptive implant that will require no surgical removal. So that means that we need to use a biodegradable implant, and optimally there would be no need for local anesthesia by reducing the diameter of the implant, or ultimately we are also working on project to develop contraceptive in the form of microneedle, which will be virtually painless by administration. So the objective of that on this product is to produce the contraceptive implant with smaller diameter, and important characteristic of the implant is that the API or the hormone in the plasma must be very constant to keep the efficacy within 6 months. And after releasing all the payload of the drug, and the plasma concentration of the drug should drop rapidly, so that the patient or woman can return to fertility if there's no need to further use contraceptive implant. And the implant should be bioresorbable, resorbable to prevent the surgical removal. So we developed the Symbiosis multi-block copolymer-based implant for contraceptive for the case of goserelin, which is a peptide, but the platform is actually available and suitable for different API which are sensitive to temperature, or which are sensitive to the decrease in the local pH. And in this case, we would also want to demonstrate that we can produce the implant using a very low extrusion temperature and low melt viscosity. And if we consider the PLGA as a reference, the melting temperature, the temperature that we typically need to use to produce a PLGA-based implant would range about 80 to 130, 150 degrees Celsius. And this would already be the degradation temperature of many APIs like protein. So when we use a Symbiosis multi-block copolymer, we can run hot melt extrusion at a temperature as low as 50 to 60 degrees. That would help us to protect the heat-sensitive API. And here is an example of the stability of a protein alkaline phosphatase by heat exposure in the oven at 55 degree. In the graph on the left side, you see that the protein remain stable after about 2 hours, 120 minutes. But when we increase the temperature to 95 degree or 130 degree, which is the typical extrusion temperature of PLGA, then we will see a rapid decrease in the activity of the protein. Using hot melt extrusion process and Symbiosis polymer, we run the hot melt extrusion at 50 degree, 55 degree, and 85 degree, and we can see that at 55 degree, using Symbiosis polymer, we can produce the implant of a protein, and we can maintain the activity of the protein at about 80%, much higher, significantly higher than when we run the hot melt extrusion at 85 or 130 degrees Celsius. Now, next to the integrity, again, here we also demonstrated that using different polymer structure, we can control release the release characteristic of the protein. In this case, that is an bovine serum albumin as a model API. And with different API, with different molecular weight on the left side, then you can see that in one polymer, using one polymer, the release characteristic vary with the molecular weight of the API itself. So with each API, we will need to address by using different polymer, by designing the polymer structure to be suitable for the requirement. So in summary, for some key feature of the Symbiosis based implant technology, that it is an excellent control over, it has excellent control over release kinetic of the API, which is a regular release pattern, and we can tailor design the polymer to meet the release duration. And it is compatible, the Symbiosis polymer is compatible with the Thermo label APIs such as protein or peptide, or even some heat sensitive small molecule. And the swallowability of the polymer, it's a very it's a real distinction for Symbiosis polymer in development of a sustained release delivery for the pH sensitive API such as protein or peptide. So I hope that I can share with you our experiences in development of the long-acting injectable implant and the current formulation platform using PLGA and the advantage of the Symbiosis polymer platform that we can use for the case of goserelin itself, but also we can use for different peptide, protein, and other small molecules that are sensitive to the temperature, and that are also sensitive to the acidic pH, especially the local acidic pH in system containing PLGA. And if you have questions, feel free to ask, or later on reach out to me. And if there's any potential idea, we are very open for discussion. And I would like to thank you a lot for your attention.
Thank you very much, Tan. Hello again, everyone. I hope you enjoyed the presentation and found it relevant and engaging. Thank you again, Tan, for the insightful talk you just delivered. We have a number of follow-up questions here for you. And for the audience, just a reminder, if you have a question that you'd like to ask, you can submit it using the Ask a Question panel beside the media player. So, Tan, the first question I have for you here: what other manufacturing techniques can be used with Symbiosis polymer besides hot melt extrusion?
That's a very good question. Thanks, Darius. Now, as I presented today in this presentation, I focus on the hot melt extrusion. But next to hot melt extrusion, we also manufacture microparticle to deliver a sustained release API. And so we manufacture by a solvent-based process, and we also manufacture or use Symbiosis in a coating application, where in one of the product that is now has been in the market for a few years, we coated a stand for delivery and API. And we are also now working on a solvent-based process to produce Symbiosis-based in situ forming gel as well.
Great, thank you. Another question for you. When working with heat-sensitive API, to what extent can you decrease hot melt extrusion processing temperature?
So up to now, the lowest temperature that we have used to produce implant is about 50 to 60 degree, and that corresponds to the selection of the monomer that we will use in the structure of multi-block copolymer. And in this case, we design in our polymer a large amount of caprolactone, polycaprolactone, and therefore that would allow us to decrease the extrusion temperature. And one thing that I would like to mention is that next to the low extrusion temperature, the Symbiosis polymer will also, when melted, has a low melt viscosity. That will also contribute to the stabilization of the heat-sensitive API.
Great. Another question: do you also sell this polymer off the shelf?
That actually I have to apologize. That's not our business model. We do not sell the polymer off the shelf. The reason behind that is that we have a different design of the polymer, and that will require understanding of the structure of the polymer to interpret data. And if we just sell the polymer off the shelf, it is quite easy to lead the user to make incorrect conclusion about the drug delivery system. So we normally work with our partner or client or partner in a very small project that we call feasibility project, where we will help the client in selection of the polymer, and also manufacturing a prototype and test it in the lab, and later on in the first preliminary animal model to show the proof of concept. And that is how we are working at Innocore using the Symbiosis polymer.
Great. And we have time for just one more question here. And how or what is the regulatory status of your Symbiosis polymer?
So the Symbiosis polymer is used, or it is produced using the very well-known monomer. Then all these monomer have excellent safety and regulatory status, and the next to the regulatory status of the monomer, we also have one of our Symbiosis polymer that has been used in a product that is now in the market for a few years, And this is also a good example of the safety and regulatory status of the polymer. So recently we have a project with a client going to a clinical stage, and we have been in discussion with the FDA together with our client about the regulatory approach for the phase one clinical study. And we've been aligned with the FDA that we don't need to produce biocompatibility data of the polymer only, but we all, as a standard process, we only need to perform a tox study for the combination of polymer and the API. That it won't have to do it anyway with any kind of product. But for the polymer itself, it has been considered as safe.
Great. Fantastic. Thank you so much, Tan, for those great answers, and of course for your presentation today. And thank you,
Um,
everyone, for joining us today to attend this webinar.
Thank
Again,
you.
the slides and a recording of today's webinar will be available soon, so look out for an email giving you access to the video recording in the near future. And before you go, we'd like for you to take a moment to provide your feedback on the survey and let us know what webinar topics you'd like to see in the future. Finally, if you still have any questions, feel free to submit them now using the Ask a Panel question or Ask a Question panel, and we'll be sure to forward them along to our presenter. In closing, thank you again for taking part in this AZO Network webinar, and we look forward to sharing science with you again in the future.